Chapter 3 will be the bacterial cell structure. This is a fairly large chapter, and we will talk about some of the aspects in a little bit less detail when we will describe the PowerPoint slides, because some of the corresponding reading material which exactly follows the PowerPoint will supplement a little bit of detail as necessary. So the first thing that is important is that the bacteria are diverse. We are skipping the 3.1. We are starting with the 3.2, which is a typical, they share some of the common features. And we are going to basically describe a typical bacterial cell and discuss the factors that determine the size and the shape of the bacterial cell. So the first thing is that they do have a variety of morphologies or structures. And first, if we just take a look at this as a very simple way, that if they are coccus, singular, cocci, plural, they are spherical. Whereas the rods and oblong cells are usually called bacillus as a singular or bacilli as plural. And also the arrangement of them is determined by the plane of division and determine whether that after division they are separated or not, whether they are together like a cluster or not, and also the size. So first, let us take a look at the cocci or the coccus spheres that can be single or can be associated in arrangement that is useful for the identification. So if there's a single cell just in isolation, we will call it as a coccus. If there are many of them spread around, the plural is cocci. So the diplococcus actually divide and remain in twos. So there will be two of them together, two coccus next to each other. On the other hand, the streptococci divide in one plane, and the streptococcus or the streptococci is always form chains. Whereas the staphylococci, these are the two very important ones. Streptococci, we're going to talk about that all the time. Staphylococci, we're going to talk about that all the time. Divide in random planes and making a grape-like structure. So this is very characteristic of staphylococci, making these grape-like structure. Whereas on the other hand, there are some other types of the coccus as well, which are the tetrads, divide in two planes, and they form a square of four cocci. These square of four cocci will divide and will form another square of four cocci. So all these four cocci is a very specific type of structure you can actually see. On the other hand, the sarcina divide in three planes instead of two and making a cubic packet of 8 cocci. So really very strange and all types of divisions that are actually seen. So here you are seeing some of the pseudo-colored electron micrograph. They are original micrographs were black and white, and then they're colored artificially. These are some of the cocci, and you can see that they are separated out. They are mostly round, whereas here you are seeing the bacilli or bacillus. They are rod-shaped in structure. And here you can see that this is S. aureus, means this is Staphylococcus aureus, and they are in these bunch of the clusters that you are seeing, that what we talked about. Other than that, you have the bacilli, the rods, and where the length to width ratio actually differ, whereas the coccobacilli is a short and wide, and they are just in between the round structure and a bacillus or a rod-shaped structure. So those are called the coccobacilli. On the other hand, there are other types of shapes are also found. The vibrios are comma-shaped, whereas the spirilla, these are actually rigid type of shape structure, rigid shape structure. So here you can see this Campylobacter. This is a spiral shape. This is a spirilla. Whereas the spirochete, on the other hand, is a flexible spiral shape structure. Right here, this is a spirochete. The difference in between that a spirilla, this structure is a static structure. So all these bacteria look like this. On the other hand, a spirochete, this structure is actually a dynamic structure. And we are going to talk about its motility at the very end of the chapter, that these contain an internal flagella by which it constantly do a corkscrew motion. So when they are doing the corkscrew motion, they actually appear to be a spiral shape like this. But if, on the other hand, if a spirochete actually dies, they will be a simply long rod-shaped structure. So that's the reason, as we say, that a spirochete is a dynamic structure. It appears to be like this spiral when they are alive, but they are not when they are dead. That's what we mean by the dynamic structure. Whereas a Campylobacter, on the other hand, these are the static structure, what means dead or alive. They will look like this shape, spiral shape, no matter what. So other than that, there are some of the bacteria which also have many different types. Here you can see the Streptomyces can form these filamentous bacteria. They are filament-like. There are some of the myxobacteria which can form these unique fruiting bodies. And remember that each of these fruiting bodies actually contain thousands of individual cells. And sometimes they can form the mycelium, network of long filaments like hyphae. Usually these words are specific for the fungi or the fungus, but sometimes some of the bacteria actually can form those as well. And then there are some group of bacteria which are pleomorphic organisms that are variable in shape. So a classic pleomorphic example will be the mycoplasma. So usually the bacteria will have a cell wall, and usually it's rigid in shape, whereas the mycoplasma doesn't have a rigid cell wall, and that's why they can change in shape. And why this pleomorphic mycoplasma is actually important, this is actually one of the only example of the bacteria which can actually pass through a 0.2 micron bacteriological filter. You can actually filter out all the bacteria with a 0.2 micron filter. They cannot pass through it, although the viruses, which are much smaller, can pass through it. But with the exception of the mycoplasma, which is pleomorphic in nature, doesn't have a rigid cell wall, and it can actually squeeze through and pass through these 0.2 micron filters, and sometimes can cause problem in animal cell culture if there is a mycoplasma infection. So after that, we are looking at these bacterial cell size, and you can just see that these are very self-explanatory. We talked about these mycoplasma. See that its size is 0.3 micron. But as they are pleomorphic, they can squeeze through this 0.2 micron filter. That's what we were talking about. So on the average, you can see that 1.1 to 1.5 micron, and wide 2 to 6 micron long as such. E. coli, these are very large, 600 by 80 micron, very large on the, so this is the typical structure of the E. coli. Approximately 2 to 6 micrometer long, 1.1 to 1.5 micrometer on an average. Very large bacteria like this E. fisheloni, that could be 600 by 80 micrometers. So you can see that they are very large. So this is just an overview of that this is a E. coli, this is a streptococcus, and see that these are the different viruses compared to a red blood cell. So just an overview that bacterial comparison size-wise, other than that much detail is not necessary. So here also there are a couple of different bacteria. You can easily see large ones, and these are some of the symbiotic bacteria, which are these white dots. They are present in there. Here you can see that the E. fisheloni, these are much, much bigger. And here you can see that these two are the Prometium, which are the protozoan, and these bacteria are much larger than these protozoal cells. Just having an overview of this that they can have the size extreme, very small to very large, even larger than the protozoa or the protists. So the cells actually want to always have a high surface to area to volume ratio, and this actually helps the bacterial cell to increase the efficiency of the nutrient uptake and diffusion of molecule within the cell. And large and odd shape may be protective mechanism, on the other hand, for the predation. So let's take a look at that when we just talk about the two different sizes. Here you can see that this is a small size, and you can see that the radius of this sphere, or this, if this is a coccus, the radius is about 1 micrometer. And the surface is 12.6 micrometers square. Volume is 4.2. So surface to volume ratio is 3. On the other hand, you can see that if it is large, you can see that surface to volume ratio falls to 1.5. So this does have some kind of disadvantage that you can see that you want a high surface to volume ratio, which is like this, which increases the efficiency of nutrient uptake and diffusion of the molecule. But sometimes, like E. fisheloni, the odd shape may be protective mechanism from the predation as well. And we have seen that some of these large bacteria, they have convoluted membranes to increase their surface area of absorption for the nutrient exchange. So after that, we are looking at this common bacterial structure. We are going to talk about the cell envelope, plasma membrane, cell wall, and then we are going to talk about the cytoplasm, nucleoid, and then we're going to see external structure, pili and flagella, how they help in different processes. So we will start one at a time. So in this section, we are starting with the plasma membrane. Now, just remember that we actually do the plasma membrane in very, very detail in the cell biology, which is the biology 8420 course. That's why we are not discussing the plasma membrane, because it is basically exactly the same, and all the phenomena of the plasma membrane, like the transport, are discussed in the cell biology. So we are only talking about the basic function and a particular phenomenon which is specific to the bacteria, which is the iron uptake by the siderophores. These are the only two things that we are talking here. So the first thing is that the cell envelope, the plasma membrane, and all of the surrounding layer extend to it. So essentially, it surrounds the cytoplasm. And they commonly include the plasma membrane. Outside the plasma membrane, there could be a cell wall. And outside that, it could have a capsule layer or a cell line layer. We are going to see that there is a gram-negative bacteria where you have a plasma membrane. Then around that, you have a periplasmic space, which contain a thin layer of peptidoglycan and another lipopolysaccharide membrane surrounding it. So we will talk about all of them at a time. So the plasma membrane essentially encloses the entire cytoplasm, which is basically the heart of the bacterial cell, and every reaction, everything goes on in here. It contains the nucleoid region, which contain the DNA material for that, and essentially everything. The plasma membrane also do help in many different kind of processes. Say, for an example, energy production as well as a DNA replication. These are specialized functions of the bacterial plasma membrane, because as being prokaryotes, the bacterial cell do not have any enclosed or membrane enclosed organelle to concentrate many of the enzymes, and they do not have a specific place to carry out. So many of the enzymes are associated with the inner leaflet of the plasma membrane, and they carry out different processes. So the most important membrane that is required for all living organism, and it is made up of a phospholipid bilayer. You have an outer phospholipid bilayer, inner phospholipid bilayer. We call this as outer leaflet and inner leaflet. So the innermost encompasses the cytoplasm, and they are selectively permeable barrier that acquires nutrients and eliminates the waste and interacts with the external environment. Now, there are many different types of protein, which are the integral or transmembrane protein. These spans the entire width of the plasma membrane. And along with that, there could be peripheral proteins, which usually are present either on the outer surface of the outer leaflet of the plasma membrane or in the inner leaflet of the plasma membrane, either only exposed to the outside environment or exposed towards the cytoplasm. And it detects and responds to surrounding chemicals. And there are many different type of transport systems, which contains channels, which contains specialized type of transporter and pumps, many different types. And also, as I said, that they are responsible for very different types of processes as well, because many of the enzymes of respiration, photosynthesis, even DNA replications, they are associated with the plasma membrane in bacteria. So let's take a look at a very specific process of the bacteria. Many bacteria actually do require iron for the building molecules, and this is required for their energy conserving process that we are going to see later part somewhere. But the ferric iron, which is Fe3 plus, is very insoluble and its uptake is very difficult for the bacteria. So what the bacteria does, that they secrete this organic molecule which is known as this siderophores. And these actually make a complex, as you can see that this is Fe3 plus. It's make a complex of this siderophore iron complex, ferric iron complex, and these the iron molecule trapped within this organic molecule can be transported inside the cell. And how this transport happens, we're not going into the detail, but there are certain type of transporter which we call as ABC transporter. These are called ATP binding cassette transporter. These are present on the plasma membrane, which can uptake these complex large molecules with the expense of ATP. So it's an energy driven process. Specialized type of transporter can take up this siderophores, which brings the iron inside the cell. So after that, we are going to go into the cell wall, and this will be an elaborate part of the bacterial cell. So the first thing that function-wise, the cell wall maintains the shape of the bacteria and it helps to protect from the osmotic lysis and toxic material, and also contribute to the pathogenicity. So the main building block of this cell wall is peptidoglycan. Also, this is known as murine. This peptidoglycan is not found in any plant cell wall or fungal cell wall or even archaeal cell wall. And this is very unique for the bacteria only. And this is a rigid structure lying outside the plasma membrane. And there are two types of bacteria based on the Gram stain that we already know. One is Gram positive, which stain purple. This has a thick peptidoglycan layer as a cell wall. And this contains a single membrane. So you have a plasma membrane. Outside that, you have a thick peptidoglycan layer. We call this as a monoderm. These are some of the new terms that is being introduced instead of a Gram positive. On the other hand, there is a Gram negative. These stain pink or red in the Gram staining. Outside the plasma membrane, they have a thin peptidoglycan layer, and this is present into a place which is called the periplasmic space. This periplasmic space is just the outer space outside the plasma membrane. And then you have the second membrane, which is the outer membrane. So as you have a plasma membrane and an outer membrane, we call these now as a diderm. These are some of the new terms that are being introduced into microbiology lately. So what are these peptidoglycan? So the peptidoglycan is a mesh-like polymer of identical subunits, and there are two of these building blocks which are associated with many different types of amino acids. So these are generally known as NAG or NAM. One is N-acetyl-glucosamine and in N-acetyl-muraminic acid. So let's take a look at this. You can see that the basic block is derived from the glucose. And as you can see that you have this 1, 2, 3, the third position, you have this CH3CH, C double bond OO minus, C, this is bonded, but this will be a COO minus. So essentially, without this, a NAM will have COOH, where it can ionize into COO minus plus H plus. So this is essentially the NAM or the muramic acid. An acid is a proton donor, so this can ionize into COO minus and H plus. H plus is the proton, so this is the N-acetyl-muramic acid. So on the other hand, the NAG is also derived from the glucose, and you can see that name is very simple. You can see that right here, this you have 1, 2, 3, 4, 5, 6, and the 1, 2, 3, 4, 5, and then 6 CH2OH. The second position, this is upside down. See that this is this way, this is this way, and the second position contain the NH or the amine group, and that's why it's an acetyl-glucosamine. So acidic group, and that's why it's an acid. The amine group, that's why it's an glucosamine. Glucose plus amine. Very simple. On the other hand, what you're seeing that they, along with these basic structures, which are covalently bonded, you can see that alternating, you will have a NAM, NAG, NAM, NAG, NAM, NAG. These will alternate. And with the NAM or the N-acetylmuraminic acid, you will have a side chain or stem. And these stem contain a couple of different amino acids. So the first one is L-alanine. The second one is a D-glutamic acid. Then you have a mesodiaminopimelic acid. Then you have a D-alanine and a D-alanine. So the interesting thing that I'm pointing out here, see this L-alanine is found in the human beings. We do have these L-alanine, and all of our amino acids, the 20 amino acids that we have, that they are all L-handed or left-handed in stereochemistry. They're all left-handed. Now see that this D-glutamic acid, mesodiaminopimelic acid, D-alanine, this is the D-handed or the right-handed, and these are not found in the human beings or in the animal kingdom. So what happens? There are not any proteases that can digest these parts easily. So that is a kind of a built-in system for the bacteria, that these structures cannot be easily digested by the proteases present in the animals or in the humans. So these, on the other hand, see that the mesodiaminopimelic acid, as soon as, if this is L-glutamic acid, yes, that is present in the other in the animals as well as the human beings. If it is an L-alanine, of course. But as soon as the handness changed from L to D, these becomes the stereochemistry doesn't allow the proteases to act upon them. And now this D-alanine at this site may form to this link peptide to one another chain, and then this D-alanine is lost. So we are going to see that how this actually works right here. So here you can see right here is an N-acetylmuramic acid. So you have this L-ala, D-glue, and then you have the DAP and D-ala. When this D-ala bonds with the next, so you have a NAG-NAM chain going this way, you have the next NAG-NAM chain, and then when this D-ala actually associated with DAP, this D-ala goes out. So this is basically the cross-linking structure. So essentially, along with that, there are another type of the structural integrity has been seen. So there are many different ways, and in gram-positive bacteria, you can see that the side chain, on the other hand, this is one type, but it can have this L-alanine, the same, and then you have aminoglucosamine, D-glue, NH2, then you have a lysine instead of DAP, and then you have a D-ala, and see that this side chain is the same thing. It's still contains 4 amino acids, but just a little bit different amino acid. And they are connected with this pentaglycine interbridge. You have 1, 2, 3, 4, 5. So pentaglycine bridge. So this is another kind of cross-linking that is actually found. So here you can see that these are some of the type of bonding that is found within the glycine 4,5 to L-alanine. And you can see that these are the different types of interbridges or the crosslinks that can be found in different types of. And again, you do not have to go into the detail of that, just an overview that how these interbridges or the crosslinks actually keep the individual strands one after another, layered wise. That is important. So that's all it is. And we are also going to see that these peptide interbridge synthesis is actually inhibited by penicillin, so that what happens is that the cell wall structure actually don't have the rigidity and it falls apart, that we are going to see in the later part. So penicillin actually works on the enzyme which synthesizes this interbridge. So on the other hand, the peptidoglycan strands have a helical shape, and the crosslink peptides for these strength. And you can see that direct crosslink connecting the carboxy group amino acid between the amino acids, And the indirect crosslinks are like this, peptide interbridges. This can form actually like this. And there are various structures that are much more in detail in your textbook. You can take a look at that as well. So now let's take a look at that, how these peptidoglycan structures are associated with the gram-positive and the gram-negative cell wall. As you can see that in the gram-positive cell wall, you have the plasma membrane and you have a thick cell wall right here. And these are much, much thicker compared to the other structure, could be even 80 nanometers, 70, 80 nanometer thick. So along with the peptidoglycan, there are some other things are available, or other things are present in the cell wall, and these are known as the teichoic acid. And these are the polymers of glycerol and help maintain the cell envelope, protect from environmental stress, and may bind to host cells to initiate infections. And there are two different types. There are some which are called wall teichoic acid, which are present only in the peptidoglycan layer, and another one is known as a lipoteichoic acid, where a part of the teichoic acid is associated with the plasma membrane and the rest are inserted into the peptidoglycan layer. So wall teichoic acid and lipoteichoic acid, and these are negatively charged as well. So here you can easily see that you can see a couple of examples of the teichoic acid. This is basically a wall teichoic acid, you can easily see. And right here is a lipoteichoic acid, where it is associated with the plasma membrane and goes to the peptidoglycan layer. So now what we are going to see, that this is basically the structure of a gram-positive cell wall. You have a plasma membrane and a thick cell wall. Now, what we are going to see that in a gram-negative bacteria, you are going to see that there is a area which is called the periplasmic space. And here's where is the periplasmic space in a gram-positive bacteria. Right here is the periplasmic space. So you have the cell wall and you have the periplasmic space. And this contains a fairly few amount of protein. And what happens that although this the structure right here, which is the peptidoglycan, seems to be rigid and cross-linked, but they do have the ability to stretch a little bit and contract, and that's the reason that they actually can help in the process when there is an osmotically unfavorable environment. The bacterial cell actually do not lyse due to the presence of these cell wall layer. Because if you just only have the plasma membrane, if there is an osmotically unfavorable environment, like in hypoosmotic or a hyperosmotic environment, the cells will either shrink or it will swell and burst. But due to the presence of that, it can maintain that. So why the periplasmic space is important? Because what happens that many of the molecules which actually move through the peptidoglycan layer first diffuse into the plasma membrane from the outer world, and then they can be transported inside the cytoplasm. And the proteins can almost a fairly large amount of proteins actually can move through the peptidoglycan layer as well. And sometimes, depending upon that, the tightening up of the cell wall can restrict some of the protein movement in as such. But there are movement of proteins, and even large molecule can diffuse through the plasma membrane. So on the other hand, when we are looking at the gram-negative cell wall, it's more complex than the gram-positive and consists of a thin peptidoglycan layer surrounded by an outer membrane. So what we have right here, you have the plasma membrane. After that, you have the periplasmic space. And think about that. In the periplasmic space, you have this thin peptidoglycan layer, which is about 10 times thicker than the gram-positive bacteria. They are basically kind of floating in the periplasmic space. This is a fluid filled area, and there are many proteins and transporters in here. And then you have an outer membrane, but this outer membrane is known as the lipopolysaccharide. The inner leaflet is made up of the usual phospholipids that you find in the plasma membrane, but the outer leaflet has different kind of proteins, which is known as the lipopolysaccharides. And of course, there are many lipoproteins as well, and they do not contain any wall teichoic acid or the lipoteichoic acid. So we will see the little bit detail of these structures. So here you can see the outer membrane outside the peptidoglycan layer. These are connected to the peptidoglycan layer with a specific type of protein, which is known as this bronze lipoprotein. So here, right here, these are the bronze lipoproteins. You can see that these are associated with that. And also the periplasmic space is different, and it constitutes about 20 to 40% of the cell volume. And many enzymes are present in the periplasm: hydrolytic enzyme, transport proteins, and other proteins. So here you can see that there are many different type of transporters. So let's take a look at a couple of them. See, right here is an ABC transporter. We talked about for the siderophores. These are ATP binding cassette transporter, can transport large molecule with the help of ATP hydrolysis. On the other hand, see that right here, there is a large outer membrane receptor which can receive large molecules, and then you have a ton B transporter. This actually helps to transport molecules into the periplasmic space, and from the periplasmic space, other kind of transporter, including ABC transporters, can bring molecules inside the cytoplasm. Also, you can see that these are the porine molecules, and there are many, many porine molecules across this lipopolysaccharide layer, and see that they are in trimers. This is a typical shape of a bacterial porine structure, although there are many. So what you are seeing here, what I'm pointing out in here, the main thing that you see that you have a membrane 1. This is your plasma membrane right here. Then you have a periplasmic space with a thin peptidoglycan layer. Then you have an outer membrane. Now, the inner leaflet of the outer membrane, see these small balls with the two tails of the hydrophobic fatty acid tails. The colors of these and the plasma membrane are the same. So these are phospholipids. These are all phospholipids. These are the phospholipids. But see, the color of the outer leaflet of the outer membrane is different because this is lipopolysaccharide, which does have a very different structure, O-specific side chain of lipopolysaccharides. So we are going to talk about those in a little bit more in detail. So LPS, or the lipopolysaccharide, is a signature of the gram-negative bacteria, and you have a structure which is called the lipid A buried in the outer membrane. You can see that these are those. Then you have this core polysaccharide, 10 sugars structure, which is joined to the lipid A. And after that, you have an O side chain or O antigen. And this is a polysaccharide that extends outside from the core. So what you are seeing that right here, there are these N-phosphorylated N-acetylglucosamine, and fatty acid chains are associated with these right here. And then you have these structures, which are these KDOs and HEP. So the HEP stands for the heptulose. On the other hand, this KDO, again, it is not needed, but these are basically 3-deoxy-D-mano to octulosonic acid or octulosonate. So basically, those are some of the specific names. You can just easily see that these are, again, just repeating, they are basically 3-deoxy-D-mano to octulosonic acid or octulosonate, as they are associated with the other molecules as well. And these are the heptulose. So these structures. And then, so this is essentially the core polysaccharide. And again, detail is not necessary. Just remember that you have the lipid A. So this lipid A is right here. Then you have this core polysaccharide and the O side chain. And there are many different, the same bacteria, say for example, E. coli, they may have many different types of these O side chains. And depending upon this O side chain, we call them as O antigen as well. They are numbered. Say for an example, you know that there is a very pathogenic E. coli known as the O157 strain. And this O157 is basically the O side chain type numbered 157. So that's the pathogenic signature of that O157 strain. That's it's all about. So here is the structure again. You have the lipid A, then you have the inner core, which is the N-acetylglucosamine, and then you have this outer core, which is the KDO and the heptuloses, and then you have this O antigen, which can be many different types. And these contributes to the negative charge on the cell surface, helps to stabilize this outer membrane structure. It's a permeability barrier and defense protection, and act as an endotoxin. So what does this mean? That when these bacteria actually are lysed or dies, these lipid A, they can interact with our immune system and they can cause some of the hyperreaction of our immune system. So these are known as an endotoxin. So that's basically what it is. So the endotoxin is the lipid A component of the lipopolysaccharide layer of the gram-negative bacteria. So that's what is important, and we will talk about that in detail in some other chapter, which is an endotoxin or an exotoxin. Exotoxin is usually secreted by the bacteria, which is a toxin synthesized inside the bacteria, released, and the purpose is a toxin. Usually these are protein-based, whereas an endotoxin is a part of the outer membrane of the gram-negative bacteria, which actually reacts with our immune system and cause inflammation, fever, chills, that kind of outcome. All right, so after that, we saw these porine molecules right here, which are in the trimers. And if we just look at the porine, that we are looking at the molecular structure from the top. And you can see that these are barrel-like structure, and there is an orange loop of protein. And remember that these are all amino acids, one after another. And these orange structures can actually maintain this hourglass shape of the structure. And right here in the middle, there is a hydrophilic channel. So this is an hourglass shaped structure. So depending upon the size of the molecule, say for example, 600 Dalton molecules, they can pass through this. And depending upon the chemical, depend upon the different chemicals, the passage of these, the passage can be constricted when they by the movement of these structures. So they can be also regulated to some extent. These the porins do not require any energy or ATP energy from the ATP to pass the molecules. These are hydrophilic channels lined by polar as well as charged amino acid, and that's the reason that they exclude hydrophobic molecules. So hydrophobic molecules which are not water soluble, they cannot pass through it. And again, as we know, that there are some channels which are known as aquaporins as well, which actually basically just transport water molecules. But along with that, they can pass many other molecules up to 600 Dalton in size, which are hydrophilic or water soluble in nature. So this is just one of these barrels from the side. Here you can see the controlling liquid, the loop which controls the diameter of this of the core, and this is basically the chain which basically contain all the amino acids which are polar or the hydrophilic, helps to maintain the watery channel. So this is OMPF. This is a typical porine, a very specific porine, and this is essentially you can see that these are the loops. So the arrows are the flat arrows, but what you are seeing, these are essentially beta sheets. That's what you are seeing, and there is just very little amount of these alpha helices structure in here. So these are the molecular structures. So the cell walls actually do help in the osmotic protection. So in the hypotonic environment, when the solute concentration outside the cell is less than the inside, water actually move into the cell and the cell will swell and it will lyse. And cell wall actually protects it from here. On the other hand, as I said, that if it is in a hypertonic environment, which is a solute concentration outside the cell is greater than inside, the water actually leaves the cell and the cytoplasm will shrivel up. So this is known as the plasmolysis. So just remember that plasmolysis does not mean the burst of a cell or the plasma membrane or the cytoplasm. Plasmolysis means the collapse of the cytoplasm when water leaves the cytoplasm or the cell in a hypertonic environment. That is called the plasmolysis, collapse of the plasma. On the other hand, see that what happens, that how we can experimentally determine that the cell wall or the peptidoglycan layer essentially protects the cells in the hypertonic as well as a hypotonic environment, mainly in the hypertonic environment from bursting, is this. So this is a bacterial cell, and it contains the outer, the peptidoglycan layer. So what you can do, that as they are growing, you can add the penicillin, and that inhibits the cell wall synthesis. So they are going to produce some structure which are known as a protoplast, which doesn't have the cell wall. Now, if we take this protoplast, which is just like the cytoplasm surrounded just by the plasma membrane, and if we transfer it to the hypotonic solution, the water will move in, it will swell, and the cell will essentially lyse. So these are the two different kind of structures that you can find. You can do this experiment with the gram-negative bacteria as well. So when we actually do this experiment with a gram-positive bacteria with just the peptidoglycan layer, we call this structure a protoplast. When we do this experiment with the gram-negative bacteria, we can do exactly the same. What will happen in there, that in the periplasmic space, the peptidoglycan will not be synthesized. So the cells will be surrounded by two membranes. You will have the plasma membrane, and then you will have the lipopolysaccharide membrane, and we call those as the spheroplast, and they will lyse in the hypotonic environment as well. And this experiment essentially demonstrates that how important this peptidoglycan layer is. So when the gram-positive cells lose the cell wall in isotonic environment, we call them protoplast. So if we put a protoplast in a hypotonic environment, as we said, that the protoplast will essentially burst. Whereas if we take a gram-negative cell, that will lose the cell wall. We can just use a penicillin, and that will interfere with the synthesis of the thin peptidoglycan layer that is present in the periplasmic space. The plasma membrane will remain, and the outer LPS layer or the outer membrane will remain as well. And these are known as the spheroplast in isotonic environment. But again, if you put the spheroplast in a hypotonic environment, they will also burst. And here, I have talked about that earlier, that the mycoplasma lack a cell wall, but the plasma membrane is more resistant to osmotic pressure. So after that, we are looking at some of the EVs, or the extracellular vesicles. And these are small membrane-bound particles, and we are just getting some of the ideas of these EVs. And these develops when a membrane buds out, pinches off, and is released from the cells. And many times these happens in a kind of a harsh environment when the environment is not favorable. And the gram-positive EVs made up of the plasma membrane surrounding a small amount of cytoplasm, what happens, a part of the cytoplasm along with the plasma membrane push through the openings of the peptidoglycan layer and can bud off. Whereas the gram-negative EVs are made up of the LPS layer containing the outer membrane surrounding the sample in the periplasm. What they can have, these are very tiny, and they can contain different types of proteins, nucleic acids, and they do have implications in some kind of infections. So what can happen, that they can carry toxins, they can carry plasmids or the part of the genome, and these can be taken up by the other bacteria, and these integration of these plasmids can confer some of the particular, we are going to talk about that in detail, but can confer some of the characteristic to those as well. But we are just started learning the role of these extracellular vesicles, or EVs, in a recent time. So they are not cells. Extracellular vesicles are not cells. They can carry some ATP but do not have the ability to conserve energy by any means, play a role in cell-to-cell interaction, genetic information between the cells, and transfer toxin molecules, as I have said. So let's take a look at what happens, that the cell envelope often includes some of the layers outside the cell wall. And what are those? So in general, what you have, that outside this is the outermost layer in the cell envelope. And in general, if they are present, they are called the glycocalyx. So the glycocalyx is a polysaccharide extension that aids in the attachment of the solid surface. So why I'm saying glycocalyx? Because the glycocalyx is the general term, which can be of two different types. The glycocalyx could be capsules, or it could be the slime layers. The capsules are the glycocalyx, which are well organized, made up of polysaccharide, and they are covalently bonded and difficult to wash away. So they are well organized glycocalyx. Whereas the slime layer also is glycocalyx. They are made up of the same polysaccharide, but they are unorganized and easily washed away, and they are not covalently bonded either. So these are the two different types of glycocalyx. Along with that, there are many bacteria that contain a layer which is an S layer. This S layer is a geometric pattern made up of protein that aid in protecting from ion and pH fluctuations, and these are arranged in a tile-like fashion. So very structured structure, and these are known as the S layers. And these are outside the cell wall, found in many bacteria, not all, but these capsules and the slime layers are implicated in many different kind of pathogenesis because they attach to the tissues, and it's difficult for them to be washed away, which can carry out an infection. So here you can see that the capsules are well organized. We have already seen these. This is a Bacillus anthracis. You can see right here, and visible in light microscope. They have a protective advantage, and they become resistant to phagocytosis, protect from desiccation, so the cells are not dehydrated, and also exclude the viruses, which mean bacterial viruses or bacteriophages, and the destruction by the detergents as well. So these are the capsules. On the other hand, similar to capsules, the slime layers, but they are unorganized and easily removed, and sometimes may facilitate motility as well. So here you can see that these are the regularly structured self-assembling layer of protein or glycoprotein. And in gram-negative bacteria, the S layers adheres to the outer membrane. Whereas in the gram-positive bacteria, they are directly associated with the cell wall, which is the peptidoglycan. And again, we have already talked about that. They protect from ion, pH fluctuation, osmotic stress, enzyme, and predation as well. Maintain the shape and the rigidity of the cell, promote adhesion, protect from host defense, and a lot of industries are very interested for their potential use in nanotechnology. So after that, these are pretty much all about the cell envelope. We talked about the cell wall, the plasma membrane briefly, then cell wall, and in gram-positive, gram-negative bacteria, and then we talked about the outer layers outside the cell wall. And now we are going inside the cytoplasm, and it is still pretty complex, and it is about 10 times more dense than water. And we are going to take a look at that, what are present in these. So you have the cytoskeleton. We are going to compare these with some of the eukaryotic cytoskeletal proteins. There are some intracytoplasmic membrane that are present in here. Inclusions, ribosome, nucleoid, and plasmids. We are going to talk about these in here. So the protoplast is essentially the plasma membrane and everything within. The cytoplasm is the material bounded by the plasma membrane. This is a very generalized definition as well. So the bacterial cytoskeleton is protein filaments that polymerize to form the functional filaments, extend to the full inner dimension of the cell. And there are many homologs of eukaryotic cytoskeletal elements that have been identified in the bacteria. So your textbook does have a entire tabulized format of many different types. Just an overview of those are necessary, and we are just only going to talk about three of them, and we are going to talk about a little bit of more detail about the cytoskeleton when we go and discuss the bacterial growth in the functional aspect. These are similar to the actin, microtubules, and intermediate filaments. These are the three different types of cytoskeletal structure that are found in the eukaryotes, and these are functions are almost similar to the eukaryotes, participate in the cell division, localize proteins, and also maintain the cell shape. So what are the three most important ones? The FTSZ essentially form a Z ring, and this Z ring forms at the center of a dividing cell, and that constrict as the daughter cells separate. Just think about that usually you find a cleavage furrow that happens in the middle of the cell when an animal cell is dividing. This cleavage furrow, that is essentially the Z ring in the bacteria, which is aided by the FTSZ. And there are many different functions are carried out. You can read about that in detail in your textbook as well. So the MREB, on the other hand, that many rod-shaped structures, and these maintain the shape and the positioning of the peptidoglycan synthesis machinery. And we're going to talk about this MREB protein as well when we talk about the cell growth and the division. And also there is another one, which is the CRES. This also maintains this curved shape of the structures. And these are some of the specific type of proteins which are known as this MBL. And you can easily see that they are present in a perpendicular, in a helical format, compared along with the length of the cell. So here you can see that they are going in 90 degrees compared to the length of the cell, and maintain the curved shape of many of the bacteria. MREB is also present, and these are all important cytoskeletal proteins. And the overview of these proteins are good enough for here, and we are going to talk about their application and mechanism, that how they help in the cell division in the later part, in a different chapter. So other than that, there are lots of intracytoplasmic membrane that are found in some of the bacteria, particularly in the photosynthetic bacteria, and these are known as the thylakoids, just as the thylakoids that are found in the plastid in photosynthetic organism. And these are also found in some of the bacteria with high respiratory activity, which essentially localizes many different respiratory enzymes, and also may be found aggregates of the spherical vesicles. And very interestingly, some of these spherical vesicles are essentially formed by a phase separation of these proteins from the cytoplasm. So they are not enclosed in a membrane, but these are just protein structures which are localized. So particularly the rubisco, the complex of the rubisco complex are found in some of these spherical vesicles in some of the specific bacteria. You're going to see these pictures in your textbook as well. On the other hand, these inclusions are formed by the aggregation of organic, inorganic substances, and the primary function is to segregate cellular components so they do not diffuse freely into the cytoplasm. And there are many different types of granules, crystals, globules of organic and inorganic material stockpiled for the cell for future use. Very common out of them is the glycogen. Then you have these polyhydroxyalkanate, which is essentially the most common of these polyhydroxyalkanates are the polyhydroxybutyrate. And essentially what these are essentially the source of carbon. So basically, essentially, food source for future use. Along with that, you can also find sulfur. Then you can see polyphosphate granules. These are some of the structures. So when they are formed in this polyhydroxyalkanate or the carbon, sometimes these are known as a carbonosomes. These inclusions are also known as carbonosomes as well. Along with that there are several different things that can present. and some are enclosed by a single layer of protein or lipid shell, and these can be referred as microcompartments, and sometimes often used to sequester enzyme that produce actually toxic intermediate so that it is not detrimental to the cell. So here is one of the interesting part is that you can see that the microcompartments are not bound by the membranes, but specific functions. These carboxysomes, on the other hand, remember that this is different than the carbonosomes. I refer to something else, that when your carbons are essentially associated in a form of polyhydroxyalkanate, but particularly polyhydroxybutyrate, those are known as the carbonosomes. These are the food source. Here, this is a carboxysomes. These are present in the CO2 fixing bacteria. And these contain the enzyme carbonic anhydrase that release the carbon dioxide into the shell so that it accumulates this high concentration. And this rubisco after that made this sugar. So here you can see that this is a structure of this rubisco, and how this compartmentalization happens in the bacteria are very interesting, that this is basically called a phase separation. They are not enclosed in a membranous structure, but the bacteria does have the ability to precipitate these proteins in a particular way and separate their phases still in perfectly working order to concentrate into these carboxysomes, which essentially do these CO2 fixing and for making sugar. So after that, also the inclusion can contain the gas vacuoles, and these gas vacuoles are involved in bacterial movement. Particularly, they help in the buoyancy of the bacteria, particularly the aquatic bacteria, at which depth they are going to be present. And why it is important is that in certain depth, they may receive optimum amount of sunlight, they may receive optimum amount of food, and depending upon that, they can make these aggregates of hollow cylindrical gas vesicles, and these are known as these gas vacuoles. These are also termed as inclusions. Also, along with that, there are some bacteria, these are called the magnetotactic bacteria, and these are found in some of the aquatic bacteria, both in the north and in the southern hemisphere. And these magnetosomes are made up of this magnetite, and they orient along with the Earth's magnetic field. And there is a MAMK. This is another cytoskeletal proteins. These are associated with these magnetites, and they are oriented in a certain directions. And why this is important? Because this actually helps to orient the bacteria at a certain way, at a certain depth. And again, these are all essentially to receive the maximum amount of nutrients. So that's also very interesting for some of the magnetotactic bacteria. So other than that, what you also have in the cytoplasm is the ribosomes, which is essentially the machinery to synthesize all the proteins that the bacteria will need. And this is a complex protein and RNA structure. And the bacterial and archaeal ribosomes are the 70S ribosome. So what happens that the bacterial ribosome does have the small subunit and the large subunit. The small subunit is made up of this 16S ribosomal RNA. And along with that, you have 21 different proteins. And the large subunit contains 23S and 5S ribosomal RNA, and contain 34 different proteins. So altogether, you have 55 different proteins that are present along with the ribosomal RNA. So what is important here is the S, and sometimes your textbook actually talks about that in detail. The S is the Svedberg unit, and it is known as the sedimentation coefficient. So the sedimentation coefficient is computed out for each of these molecules when they are centrifused in an analytical ultracentrifugation. So this is the speed by which these molecules actually precipitate. So this essentially depends upon the molecular weight, but not only solely on the molecular weight, also depending the volume and the shape. So not only molecular weight, but along with other parameters, as I said, the volume and the shape. And the larger the Svedberg unit is, the larger usually the molecule is, or the heavier that molecule is. That means that will precipitate faster. So if we just say that a 16S ribosomal RNA, this will precipitate slower than the 23S. So out of 16S, 23S, and 5S, if we just think about that, the 23S ribosomal RNA will precipitate fastest in this analytical centrifugation, then the 16S RNA and the 5S. But if you take a 70S ribosome, the entire ribosome, that will sediment the fastest. So this is essentially gives you an overview of the size, volume, and the molecular weight of the structure. So that's all about the ribosomes. The nucleoid is essentially not a specific structure, but it is a region, and usually it's not bound by the membrane, and it's a location of the chromosome and the associated protein. So essentially, this is the area where the DNA of the bacteria is present. And usually the bacterial DNA is one closed circular DNA, and sometimes there are supercoiling, and there are some of the nucleoid proteins which aid in folding this structure. Remember that in eukaryotes, you have these histone proteins which form these nucleosomes. Those histone proteins are not present in the bacteria, but there are much simpler types of some of the proteins are associated, which are called the nuclear associated proteins, or NAS. These interact with the chromosome. The reason for that, the chromosome must be compacted to accommodate it inside the bacteria. Say, for example, for an E. coli genome, if we just open up the E. coli chromosome, it will be probably really very, very long. It will be probably 10 times the size of the bacterial of the bacterial cells, or even more. So it needs to be supercoiled, and these are some of the proteins which are called the nucleoid proteins, help in supercoiling those DNA into the nucleoid region. So nucleoid region is not a nucleus, not membrane bound, but where the DNA is essentially found. On the other hand, the plasmid are the extra chromosomal DNA. These are essentially the round, double-stranded closed circular DNA molecules. They exist and replicate independent of the chromosome. And some of them are called the episomes, which are able to sometimes integrate into the chromosome, and they do have the ability to excise them out of the chromosome and be present in a separate entity. And these are inherited during the cell division, and they actually do carry individual genes and contain a selective advantage in some of the situations. Often, many of the virulence genes of some of the bacteria which contain toxin genes, or some of the genes which are responsible for evasion of the immune defense of the host. These are often found in some of these plasmids. And very interestingly, that how, in a practical purpose, that how this can happen. Say, for an example, you have a rogue E. coli, you are just getting an infection of, which contain a plasmid which contain this toxin producing genes, which causes the diarrhea. Now, your own colon do have in the gut, you have plenty of these E. coli which are naive, which produces vitamin K, help you out, help in the digestion, doesn't cause any problem. But in an infection, what can happen that many of these plasmids can be transferred to some of these naive E. coli's, which now can produce this toxin and quickly surmount or attack of a rogue E. coli. So you can see that your own E. coli's can be transformed by these rogue E. coli, which is coming as an infection, and in a very quick fashion, this can escalate into a big infection. So these are some of the advantages of these plasmids, or the strategies that how the plasmids actually work in the bacterial life cycle in real life. So after that, we are looking at some of the structure, external structure for attachment and motility. So these are two types. These extend beyond the cell envelope in the bacteria, and they function in protection, attachment, horizontal gene transfer, cell movement, and one group of them is the pili or the fimbriae. Fimbriae singular or the fimbriae plural, and also the flagella. So first we are looking at the fimbria singular, and fimbriae is the plural, or pilus singular or pili plural. So these are the short hair-like thin structures, and could be present about 1,000 per cell, and can mediate the attachment. So here you can see that these are the fimbriae, and these are aid in the attachment to attach the bacteria on a substrate. On the other hand, there are these pilis, and there are certain types of pilis which are known as the sex pilis. So here you can see that these are longer, thicker, and less numerous rod. These are encoded by specific plasmid. That means that a bacteria, if it doesn't have the plasmid, they does not have the ability to produce this pilus or pili. These required for conjugation. What it means that this bacteria can be associated with another bacteria, and they can connect by this sex pilus, and a part of its chromosomal DNA or a plasmid can be carried through this to confer the genetic material to the other. So think about that. This conjugation process by the help of the sex pili is a kind of a sexual reproduction, quote unquote, sexual reproduction for the bacteria, essentially for genetic information change, because bacteria divide by the binary fission 1 into 2, but one bacteria can exchange part of its chromosome, not the whole, because it is too long to go through, but a part of its chromosome, but an entire plasmid or multiple plasmid can be transferred, because they are much smaller, through the sex pilus to another organism to confer the traits so suppose a bacteria does not have a trait, whether the bacteria does have a trait. Usually we call that the bacteria does have a trait as a F plus. Another bacteria which doesn't have that trait is F minus. So this F plus can confer or transfer its own plasmid through the sex pilus to the F minus. Now the F minus becomes F plus. Now it can transfer its F plus plasmid to an F minus, and so on. On the other hand, the flagellas are the thread-like locomotor appendages extending outward from the plasma membrane. Motility, sometimes attachment to the purposes, sometimes they are virulence factor. But the function, the motility is the main function for this bacteria. Depending upon where you have the distribution of the flagella, if you have one flagella, we call this a monotrichous. The polar flagellum is usually at the end. If you have like this, that end, this is a polar monotrichous flagella. Amphitrichous means that both end, they do have this flagella. Lophotrichous essentially is that if you have a kind of a bunch of flagella at one end, we usually call this as a lophotrichous. So this is a lophotrichous, this is a lophotrichous. This is, on the other hand, is amphitrichous, because they have both end. And the peritrichous flagella is spread entire surface of the cell. So depending upon that, what they are. So the bacterial flagella is a very unique structure, and this is a thin, rigid protein structure and cannot be observed with a bright field microscope unless specifically stained. And we talked about that how we use the potassium alum and the tannic acid to increase the size of it, and then we can stain it to visualize that, but readily visible under the electron microscope. What they have a filament, then they have a hook. This is a short curved segment, and then do have a basal body. So their structures are essentially different for the gram-positive and the gram-negative bacteria. So the gram-positive structure is straightforward, where you have the basal body made up of two rings, which are embedded, one in the plasma membrane, and another one is essentially in the peptidoglycan layer, or in the periplasmic space and the peptidoglycan layer, just made up of two rings. On the other hand, for the gram-negative rods, or the gram-negative bacterial flagella, they are a little bit different. You have an L ring, and they are named just where they are. L ring is present in the lipopolysaccharide layer. Then you have a P ring. This is associated with the periplasmic space, you can see that the periplasmic space, you have the thin peptidoglycan layer. So it's associated in the peptidoglycan layer of the periplasmic space. That's why it's P. Then you have an MS ring right here. And these are the plasma membrane. And then you have this C ring, which is on the cytoplasmic side of the plasma membrane. Cytoplasmic, then plasma membrane, peptidoglycan in the periplasmic space, and the lipopolysaccharide. 4 rings are present in there. So along with that, there are several different types of proteins associated with that, which essentially gives it a motor protein ability. So here you can see that this MS ring is known as a protein known as a FlyF. And there are many different proteins called FlyG, FlyN, and then you have this mode B and mode A. This essentially works just like a stator mechanism. So essentially what happens that you can see this, there is H plus ion moving down the concentration gradient from the periplasmic space to the cytoplasm. So what happens, the movement of this is essentially done by the movement of these proteins, or how these proton motive force essentially drive them. Remember that the flagella is not driven by the ATP, but it is driven by what is called the proton motive force. So it's a movement of the H plus ion. So there are many different enzymes which are present on the plasma membrane. They pump out H plus, or the proton, into the periplasmic space. And then when required, this high concentration of H plus, or the proton, falls back through these proteins, which are essentially the FlyF, mod B, mod A proteins in between them. They fall through this, and as they are going down the concentration gradient, or as the protons going down the concentration gradient, they actually move the flagellar motor. So essentially PMF, or proton motive force, drive this flagella. So the synthesis of flagella is very interesting. The structure is actually hollow. So here what you are seeing in the picture, that a ribosome is reading an mRNA and a protein is being synthesized. These are called the flagellin proteins. And these flagellin proteins are transported straight through the hollow membrane on the tip of the filament of the flagella, where there is a filament cap protein. And this filament cap protein essentially allows the flagellin to properly precipitate and form a rigid structure. And this is completely done by the self-assembly with the help of filament cap at the tip. So essentially, the tip of the flagella actually grows. It is not that it grows from the base. So what is the difference? Remember that for the sex pili, or the fimbriae, they grow from the base. So the units are added. So say for example, for the pilus, the pilin is the protein, which are added at the base, and they go increase in the size or increase in the length. Whereas for the flagella, they grow in length from its tip. So this is the basic difference. So these are some of the different type of motility, and these are very this portion is very self-explanatory, and we will go quickly over through that. And again, much detail is not necessary for that. So the flagellar movement allows them to swim. And then there is also another movement that is found, which is called a swarming. And usually the swarming is at the center of a Petri plate. The bacteria start, and usually they have the peritrichous flagella. That means that they have the flagella all around. And the group of these bacteria actually spread with a particular pattern, swarm, and form these typical shapes. So this is basically the swarming. And after that, we are going to see a spirochete motility and essentially a couple of different things. So the twitching and the gliding motility is also essentially by the specific type of movement of the flagella. And the chemotaxis is essentially the bacterial movement towards a source of a preferred chemical or away from it as well. And the spirochete motility we are going to talk about in a little bit more detail. So here we are seeing some of the swimming, some of the swimming abilities. So this is a forward run, very rapid rotation. You can see that these flagella can rotate at 1100 revolutions per second. And in general, there is a counterclockwise rotation that causes a forward motion, which is a run, whereas a clockwise rotation essentially disrupts the run and it causes a tumble so that it just goes upside down, upside down, upside down. And you can see that these are the different types depending upon the flagella. So this is a monotrichous polar flagella, whereas here it's all over the place. This is essentially, can think about that this is a peritrichous flagella all over the place, and they are all facing to, and when they are going anti-clockwise, they have a forward run, but when they go in the clockwise direction, then they tumble. These type of movements are found. So here is that already I talked about, that flagella is a two-part motor producing the torque. The rotor is the moving parts, and this is made up of this C or the fly-G protein. The ring turns and interact with the stator. The stator is the stationary part and forms a channel through the plasma membrane. And these are with the help of this MOT A and the MOT B channels that we just saw, this MOT A and the MOT B channels. So essentially, this is the rotor, and this MOT A and the MOT B is essentially the stator. So, when the protons move through these MOT A channels using the energy of the proton motive source, and the torque powers the rotation of the basal body and the filament. So this is the swarming. We already talked about that. When the flagella move in unison across the moist surfaces, most swarmers have the peritrichous flagella and produce a molecule that lowers the surface tension, and that's why they can spread across in this way. Now, this is an important important consideration, which is the spirochete motility. And there are several types of pathogens which are spirochetes. Say, for example, we had a picture of the Borrelia burgdorferi. The Borrelia burgdorferi is essentially causes the Lyme disease, which can come from the deer tick as such. On the other hand, Treponema pallidum, which actually causes the syphilis, essentially. That Treponema pallidum is also a spirochete, and their spiral motion actually helps in to dig through the tissue. So they can penetrate the tissue by these spirochete type of corkscrew motion motility. So what they have, that they undulate the entire cell, and their flagella is the periplasmic flagella or an internal flagella. So see that the hook and the basal body is associated with the membrane, and the filament runs inside wraps around the bacterial cell, in the periplasmic space. So the flagella remains inside the cell wall, and the rotation of this corkscrew shape exhibit the flexing and the spinning movement, and that is essentially the spirochete motility. So this particular flagella, which is also known as the periplasmic flagella, is also known as an axial filament, or axial flagella. Along with the periplasmic flagella, this is another word that you may encounter. And we already talked about this twitching, and the gliding motility occurs on solid surface, do not involve flagella, sometimes may contain these type 4 villi and the slime. And the twitching is essentially for the pili at the end of the cell, short, intermittent, jerky motion, and the cells are in contact with each other. And the gliding is the smooth movement and do not require any kind of appendages whatsoever. So chemotaxis, on the other hand, is a movement towards a chemical attractant or away from a chemical repellent. And there are many different types of receptors which are present on the envelope of the bacteria, which can essentially trigger these movements. So these bind to the chemoreceptors that transmit the signals through the chemosensing system. Very sophisticated system, even in the bacteria. In presence of the attractant repellent, the tumbling frequency is reduced. Runs towards or away from the compounds are usually longer. And essentially, the bacterial the behavior of the bacteria can be altered by temporal concentration of the chemicals. So essentially, just think about that if you have from lower to higher concentration of a gradient, it will be seen the bacteria when moving towards that, the speed of the movement of the bacteria will increase with the concentration of the chemoattractant. So the last part, we are going to talk about the bacterial endospores, and these are essentially very unique structures which do have implications in pathogenesis, but they are essentially a survival strategy for the bacteria. So the bacterial endospores are complex and dormant structure, and they are formed in response to the nutrient depletion. So when there is a harsh environment which is not favorable for growth for the bacteria, they actually switch into this endospore formation. And they are resistant to heat, ultraviolet, gamma radiation, chemical disinfectants, and desiccation. You can only use autoclaving to essentially destroy these endospores. So very difficult to destroy them. So there could be many different types of endospores. So you can see that it can be centrally produced, subterminal, terminal, or sporangium, depending upon that. But essentially, how is the structure? So you can see that the structure essentially is the core, and the core contains the genetic material and the enzyme required for the endospore to return to its vegetative cells, or the vegetative cells are the cells. Remember that this word is very important. Vegetative. So sometimes when a person actually goes into a coma or something, we call it as a vegetative state. Do not confuse this vegetative state as a dormant state. The endospore condition is not do not use the word vegetative. This is an endospore condition. A vegetative cell is an active cell which is dividing in normal condition. A normal cell, that is a vegetative bacterial cell. So just make sure that the wording here could be a little bit confusing. So that's why I explained that, because earlier some students raised concern about that understanding of the word. A vegetative cell is a live cell which is actively replicating and doing all its function in a favorable environment. When the condition is not optimum, at that time, a vegetative cell will essentially form an endospore by the process of sporulation, and the endospore will remain as long as it's required in the harsh environment. When the favorable condition returns, the spore will germinate into a vegetative cell to return back to its normal life cycle. So there's a core, then you have an inner membrane, and after that you have a germ cell wall, then you have a cortex layer, cortical layer, and you have an outer membrane, and you may or may not have an exosporium outside that. So essentially, the cortex beneath the coat has a thick peptidoglycan, which could be almost 100 times thicker than the bacterial cell wall. And that's one of the reasons which gives this huge rigidity. There are some other factors as well. The core has the nucleoid and the ribosomes, and we're going to see that the nucleoid, the DNA is actually compacted in a certain way and get associated with other like a calcium and dipicolonic acid and dehydrated, so that they are not destroyed or they are not susceptible to ultraviolet radiation. It has been found that even some of the spores, which are 250 million years old, can be germinated as well. So you can easily imagine that the scientists have found endospores 250 million years ago from those and could germinate them. So the sporulation is the endospore formation process, and organized process takes several hours, starts when the growth slows due to a lack of nutrients, and produce a dormant cell that can persist until a nutrient. So a dormant cell is essentially the cell which does not do any form. any function of its normal, whereas a vegetative cell is an active cell. So I'm just again re-emphasizing these two terms, vegetative cell versus the dormant cell. So here you have a cell division, and this cell division essentially becomes unequal when a sporulation happens. And these are some of the axial filaments are formed, and you form a structure which we call as a four-spore structure is formed. And this septum, essentially the plasma membrane, unequally divides the two compartments, and this is the septum formation or the four-spore formation. And then the larger part, the plasma membrane, engulf the entire part of the four-spore, and it is enclosed in a double membranous structure. At that time, you can easily see that there are several changes that happens with the DNA. As I have said, that the calcium as dipicolonic acid get associated with the DNA. There are several different chemicals are intercalated, and the DNA actually converts itself from the B form of the DNA to the A form of the DNA. And the A form of the DNA is much more resistant to ultraviolet radiation and other environmental damages. And at this stage, this cortex formation happens with all the enzymes. So the peptidoglycan layer is being laid down in here. And here you can see the thick peptidoglycan cortex is essentially formed, and the coat is essentially being synthesized. And this slimy exosporium can be outside. Sometimes always the exosporium is not there, but this is essentially a layer which can help them to stick to certain structures or help them or prevent them from too much desiccation as well. But again, the structure with or without the exosporium is still very, very stable in the environment. And what happens that the sporangium right here at this point, which essentially made the endospore, essentially lies. And so essentially the cell that actually produced get destroyed, and the free endospore goes into the environment. Many of the bacteria produce these endospores. So out of those, the one of group of bacteria which are anaerobic bacteria, Clostridium, essentially produce these spores. So say for an example, Clostridium botulinum, which causes the botulism, one of the most deadliest toxin that is known to man, and that causes the gastrointestinal disorder, food poisoning. Then you have a Clostridium perfringens, which can cause gas gangrene as well. And then you have a Clostridium tetani, which causes a tetanus. All of them can form these spores. Even some of the extremely dangerous pathogens like Bacillus anthracis that can form endospores as well. And they can be spread into the environment, and they are very difficult to destroy until certain types of endospore, certain type of chemicals or autoclaving is actually performed. So just regular cooking and heating does not destroy this endospore. So when favorable conditions actually return, gradually this absorbs water, and some of the enzymes are activated, and those essentially start the vegetative process of the cell. Means that all the enzymes essentially start working back again, and this essentially germinate, or the spore essentially germinate, which will form again the natural healthy cell, vegetative cell, which will undergo its function in a normal environment. So the endospore will remain in a harsh environment, which is not favorable, as long as it is needed. It is the survival procedure. And prepares endospore for germination. The germination process, receptors detect the small molecules, that is sugars, amino acids, and the emergence of the vegetative cells. So essentially, that is all about this chapter. It's a pretty big chapter. So essentially, remember that bacteria have many different structures, and there are some fundamental items that are present virtually in all of them. And we will continue to examine the two domains of life. And after that, we will follow the structure of the archaea. And always remember that the structure always determines the function. So thinking about how different cellular structures, bacterial in this case, give the cells a particular function for their particular environment or the needs.